So, good evening, everyone, and this is your final session. So, this will be probably a revision session in which we'll go through all the topics that you have looked into in the entire 12-week sessions. So, let's start. So, firstly, we'll start with week 1, and one by one, we'll go through all the week, means from week 1 to week 12, all the MCQs that we have practiced, and along with that, we'll also look into the things which will make each and every concept clear so that you will be ready for the exams. So, in week 1, you have looked into the introduction to genes and genome organization, and you've also looked into the history and basics of genetic engineering. Along with that, you've looked into advantages and limitations of genetic engineering. So, let's start with the first MCQ. So, the first question is, who coined the term genome? Option A, Frederick Griffith. Option B, Francis Crick. Option C, Wilhelm Ludwig Johansen. And option D is Hans Winkler. So you can pause the video and think of the right answer. Okay, so the right answer is option D, that is Hans Winkler. But we'll also look into the other three scientists as well. So if we talk about the Frederick Griffith, so he discovered the phenomena of bacterial transformation, whereas Francis Crick, so he co-discovered the double helical structure of DNA along with the Watson, which is later on known as the Watson and Crick model of DNA. Then Wilhelm Ludwig Johansen, so he coined the term gene, genotype, phenotype, but not genome. So when we say genome is the complete set of all the genes which are present in the organism at a particular point of time, that is known as genome. So Hans Winkler is the correct answer because he coined the term genome in 1920 by combining gene and chromosome. So that combines up to genome. Okay, let's move to the next question. So the next question is, for the 64 codons, which of the following statements are true? Option A, 61 codons code for the amino acids and the remaining 3 are the stop codons. Option B is 60 codons code for amino acids and the remaining 4 are the stop codons. Option C is 63 codons code for the amino acids and the remaining 1 is a stop codon. And option D is 62 codons code for amino acids and the remaining 2 are the stop codons. So you need to tell the correct statement among these 4. So do you want to make a guess, Vivina? Okay, then let's see. So the right answer is option A. Yeah, you're right. So right answer is option A, that is 61 codons code for the amino acids and the remaining 3 are the stop codons. And here is the codon table. So those 3 stop codons are UAA, UAG and UGA. So these do not code for any amino acid and the remaining 61 are the ones which code for the amino acids. Okay, so our next question is, histone DNA interactions are mediated by what type of forces? Option A, water mediated hydrogen bonds. Option B, ionic interactions. Option C, non-polar interactions. And option D is all of the above. Okay, so let's see. So right answer is option D, that is all the above. So all these interactions are involved in the histone and DNA interaction. So first one was water mediated hydrogen bonds, and why these are required? Because these helps the stabilization of histone and DNA complex, and especially between the DNA bases and histone residues by the water bridges. Then second one was ionic interactions. So this is the primary force of interaction between the DNA and the histone, because DNA is negatively charged, because DNA contains the phosphate group due to which the DNA has the negative charge. So it interacts strongly with the positively charged lysine and arginine residues which are present in the histone. So these are the positively charged amino acids. So due to the opposite charges, there is an ionic interaction between them, and this is the primary force of interaction. Then third one was non-polar interactions. So it contributes to the histone core stability and histone DNA packing through the hydrophobic effects. So there are certain amino acids which are present in the histone which are hydrophobic in nature, and they interacts with the DNA. So these are the known polar interactions, and all of them, they helps the stabilization between the histone and DNA and let it maintain that structure. So means when you look at the eukaryotic genome, so you might have seen that the eukaryotic genome has a DNA which is compactly packed. So how it is packed? With the help of histone. DNA is wrapped around the histone proteins and they adopt a compact structure, due to which we see a structure called as nucleosome, and that nucleosome is again wrapped and it make a again more compact structure which is chromosome, which you see. So generally DNA is a thin thread like structure which is very long. So to make it compact and arrange it inside the nucleus, this folding is very important, which takes place with the help of histone, due to which the DNA is able to wrap around it and adopt a compact structure. So next question is, the first type 2 restriction enzyme to be identified was option A BAMH1, option 2 EcoR1, option 3 means option C HIND2, and option D is TAC1. Okay, so now let's see what is the right answer. So right answer is option C, that is HIND2. So HIND2 was the first type 2 restriction enzyme to be discovered and characterized. It was identified in late 1960s, and the organism from which it was identified, so the organism was Haemophilus influenzae. So this is how the enzymes are named. So the organism from which it is obtained, so firstly the first name is kept and then the species name is kept in the small letters, so first three letters from there. uh, okay, so then the 2 specifies the serial number, that when it was sequenced, so at what serial number it was sequenced, so that's why it got the name HIND2. And mainly nowadays you'll see HIND3, which is generally used. So this was the first type 2 restriction enzyme, and this was the first enzyme which was shown to cut the DNA at specific internal sites, laying the foundation for modern genetic engineering. So generally, restriction enzymes come under the category of nucleases. So nucleases are the enzyme which cleaves the nucleic acids, and then there is another category of nucleases which is endonucleases. So nucleases can also be divided into endonucleases and exonucleases. So endo means when something some enzyme is cutting the nucleic acid from in between, from middle, so that is endonuclease. When it is cutting from the terminal, so that enzyme is known as exonuclease. So it is cleaving nucleic acids but from external side, so that is exonuclease. So these restriction enzymes are the endonucleases, and then these are specific endonucleases. That means these have the property to recognize a sequence and then cleave it. So that's why these are the special type of endonucleases. That's why they have got the name restriction endonucleases, and this was the first restriction endonuclease. And then there are another endonucleases which are BAMH1. So this is also the type 2 restriction enzyme, and it is obtained from the organism Bacillus amyloliquefaciens, and it was discovered later, means later than this. Then next was the EcoR1, which is most well known. This is also well known type 2 enzyme, and it is obtained from the E. coli, and it was discovered after HIND2. Similarly, TAC1, which is thermostable type 2 restriction enzyme, and it is obtained from the Thermus aquaticus. It is used in the molecular biology, so you might have heard its name when you talk about the PCR, because PCR takes place in the high temperature. So this is the enzyme. This is a type of restriction enzyme which is generally used because it is thermostable. Okay, let's move to the next question. So next question is, the DNA from which two organisms did Paul Berg use to create the first recombinant DNA? Option A, Agrobacterium tumefaciens and lambda-phage virus. Option B, simian virus 40 and lambda-phage virus. Option C, Bacillus thuringiensis and E. coli. And option D is hepatitis B virus and E. coli. So anyone wants to guess this answer? Okay, so you mentioned option C. So let's see. So the right answer for this question is option B, which is the simian virus 40 and the lambda virus. So Paul Berg in 1972, he created this first recombinant DNA molecule, So simian virus is the virus which is a monkey virus. So it was used as a vector, and lambda phage virus is a bacterial, means bacteriophage virus. So it's a bacteriophage, the virus which infects the bacteria. So that infects E. coli. So these two virus, means these two vectors were used, and this pioneering work laid the foundation for genetic engineering, and due to which the Paul Erg also got Nobel Prize in chemistry in 1980s for this particular discovery of the recombinant DNA molecule. So let's move to another question. So the question is, tumor inducing plasmids are derived from which organism? Option A, Bacillus amyloliquefaciens. Option B, Streptomyces griseus. Option C, Agrobacterium tumefaciens. And option D is Escherichia coli. Okay, so you have mentioned option C, that is right, which is Agrobacterium tumefaciens, and this is how the plasmid is isolated from the Agrobacterium tumefaciens. So it contains the tDNA region, and this is known as the Ti plasmid. And the name why it has got the Ti plasmid name, because it is tumor inducing. So generally it is inserted into the cellular DNA of the plant, and this is our gene of interest. So as soon as it is incorporated into the cellular DNA, then it is entered into the E. coli or any organism. So it is transformed recombinant DNAs back into the Agrobacterium tumefaciens. So in this case, any bacterial host is used, and then this DNA is inserted. So later on, this contains the Ti plasmid, and it infects the plant cells with the Agrobacterium tumefaciens, which due to which it results in a recombinant plant. So this host contains the recombinant DNA. It is uh, allowed to infect that plant, and with that it transfer its recombinant DNA into the plant. So now our plant is the recombinant plant, and whenever some insects eat that plant, so now this particular recombinant DNA will enter into the gut of that organism, and it will lead to the tumor. So it leads to the tumor induction in that organism, due to which its name is tumor inducing plasmid. Okay, so the next question is, which enzyme catalyzes the formation of a phosphodiester bond at the single strand break? Option A, phosphodiesterase. Option B, DNA ligase. Option C, polynucleotide kinase. And option D is alkaline phosphatase. So you need to tell which helps in the formation of phosphodiester bond after the single strand break. Okay, so you're right. That's DNA ligase. And to confuse you, the phosphodiesterase name is kept. So do not get confused whenever ase is written. So that means it leads to the degradation. So phosphodiesterase means the enzyme which will break the phosphodiester bond. Okay, so the DNA ligase is the right answer. And whenever we read the replication, transcription, then this ligase name comes many times, and even in DNA repair also. So DNA ligase is the one which makes the phosphodiester bond after the single strand breaks takes place, and this is how it helps. With the help of ATP, it creates the phosphodiester bond. And phosphodiester bond is the bond which is present between the sugar and the phosphate group, and that is how the backbone of DNA is arranged. So between two sugars there is a bond with the help of phosphorus, that is phosphodiester bond. Yeah, okay, so then let's talk about the C, which is polynucleotide kinase. So whenever kinase is kept, that means which adds the phosphate group. So polynucleotide kinase will add the phosphate group to the 5' end of the DNA or RNA. And then there is alkaline phosphatase. So this particular enzyme phosphatase means it is removing the phosphate group from the DNA or RNA, so it prevents the ligation. Okay, so now let's move to next question. So the question is, the genome of the lambda phage consists of option A, double stranded linear DNA, option B, single stranded linear DNA, option C, single stranded linear RNA, and option D is double stranded circular DNA. Okay, so let's see the right answer. So right answer is option A, that is double stranded linear DNA. So this is the linear DNA which later on gets circularized with the help of these cos sites which are present, but normally it is linear DNA and it is double stranded in nature. So rest other options are incorrect, whereas this okay, let's look at other options also why they are incorrect. So second option was single stranded linear DNA. So lambda phage, because it is double stranded, so that's why this option is not right. Then third option is also, it says single stranded, so it is not single stranded, and then it says RNA, so it is DNA, not RNA. Third option was double stranded, but it says circular DNA. So generally, means later on whenever you see during the, means in the recombinant DNA technology, when we use this phage vector, so in that case it is generally used in its circular form, but in its native form it is in linear shape. So that's why we'll say that the right answer is double stranded and the linear DNA. Okay, so the next question is, who coined the term replication fork? Option A, John Keynes. Option B, Maurice Wilkins. Option C, Albrecht Kossel. And option D is Rosalind Franklin. Okay, so the replication fork is the term which comes when we talk about the replication. Okay, so I mentioned option A. Yeah, it's right. So John Keynes was the one who coined the term replication fork after the visualization of structure of replicating DNA using the autoradiography in E. coli, which provided the direct evidence of DNA replication intermediates. So replication fork is like a fork-like structure which appears. So why it's fork-like? Because during replication we need to unwind the double helical structure of DNA so that we can start duplication of that particular DNA on both the strands. So due to that unwinding between both the strands of the DNA, it adopts a structure, it opens up from middle, due to which it appears like a replication, it appears like a fork. And because it takes place during the process of replication, so it got its name as replication fork. And then if we talk about Maurice Wilkins, so he worked on X-ray diffraction studies of DNA, and he also shared the Nobel Prize with Watson and Crick, but he did not coin this term. Then Albrecht Kossel, so he discovered 5 nucleic acid bases and histones, but was not involved in naming the replication structures. Then Rosalind Franklin, so she is well known for the discovery of X-ray diffraction data related to DNA, which later on helped Watson and Crick to, means, uncover the main means the structure of double helical structure of DNA. So means she was the one who started the study, but she was not able to interpret it. So later on, Watson and Crick, with her X-ray diffraction data, they were later on able to make more sense that how the DNA is arranged in the double helical form. So let's move to next question. So the question is, the flavor saver TM tomato has an increased shelf life due to the suppression of which gene? Option A, ACC synthase gene. Option B, beta galactosidase gene. Option C, sucrose synthase gene. And option D is polygalactouronase gene. So in this case, they have asked that the gene which is involved in the increase of the shelf life of this particular tomato. Okay, so you mentioned option A. So in this case, the right answer is option D, which is polygalactouronase gene. So now let's see what ACC gene synthase gene does. So it is involved in the ethylase biosynthesis, which affects ripening but was not the target in the case of this flavor saver tomato. If we talk about beta galactosidase gene, so this is involved in the carbohydrate metabolism and it is not linked to the ripening of the plant or softening of this tomato. Then comes sucrose synthase gene. So its role is in the sugar metabolism. It's not linked to the shelf life. But if we talk about polygalactouronase gene, so in the this particular tomato flavor saver, so it has increased the shelf life of this particular plant due to the antisense suppression of the polygalactouronase gene, which slows the pectin degradation and thus delays the softening of the tomato. And then one more thing which you can note here, that in this it says that the antisense suppression. So generally antisense suppression means that you use the means mRNA, which is known as the miRNA actually, which inhibits the translation of that particular RNA, so that miRNA will bind to the mRNA of this particular gene, and it will not allow that mRNA to further get translated into a protein. So that is how the gene is um, suppressed in this case. Okay, so next question is, who coined the term gene? Option A, Gregor Mendel. Option B, Wilhelm Johansen. Option C, Watson and Crick. And option D is Alfred Hershey. So this is quite straightforward. So the right answer is option B, which is Wilhelm Johansen. So he was the one who, yes, so he was the one who coined the term gene. Okay, next question is, histones are found in option A, eukaryotes, option B, prokaryotes, option C, both, and option D, plants. Yeah, you're right. So histones are found on in eukaryotes. So last session, people were confused. They mentioned that in prokaryotes also we have histone-like proteins. So those proteins are histone-like, but they are not histones. So to which the right answer is option A, which are eukaryotes. And these proteins are generally basic proteins, and these are found in eukaryotic cells, and they help the DNA to pack compactly in the cell, and the structure which it adopts is known as nucleosome, in which the DNA is wrapped around these histone proteins, and this is how the histone octamer is arranged. So means multiple histone subunits are joined together. So this is H3, H4, H2A, H2B. These combine and make a octamer like structure, and then the DNA wraps around it. And then there is a linker histone protein also, H1, which links between two such histone proteins. And in prokaryotes, they do not have histones, but they have some other proteins which appear like histone proteins. Okay, so then comes plants. So plant is also incorrect, because not just plants, because eukaryotes option was already present, so we cannot tick plants. Otherwise, plants come under eukaryotes and they have histones. Okay, so next question is, prokaryotic chromosomes are usually option A, both linear, option B, both circular, option C, prokaryotic circular, eukaryotic linear, and option D is prokaryotic linear and eukaryotic circular. So in this they have asked about both the, means the question doesn't say that, but in this, because the option A and B are like both, so in this you have to answer in such a way that you are talking about both the prokaryotic and eukaryotic chromosomes. Yeah, so the right answer is option C, that is prokaryotic chromosome is circular and eukaryotic is linear. And one more point which I want to add is that in eukaryotes, if you see the mitochondria or chloroplast, so they contains the genetic material of their own. So their genetic material is generally circular in nature. So previously there was a hypothesis known as endosymbiont theory, which believes that in the when the evolution was taking place, then the prokaryotic organism engulfed another prokaryotic organism. So they believe that these mitochondria, chloroplast, they are like the prokaryotic organisms which were eaten up by the eukaryotic organisms. That's why their genetic material resembles the prokaryotic organisms. But for this particular question, the right answer is option C. So as you can see, the prokaryotic chromosome is circular in nature, and in eukaryotes, the uh, DNA is like a linear in nature, so it is linearly arranged. So next question is, first recombinant DNA molecule was created by option A, Herbert Boyer, option B, Stanley Cohen, option C, Paul Berg, and option D is Warner Arbor. Okay, so the right answer is option C, Paul Berg, because previously also we have seen that he used two organisms, which was simian virus 40 and lambda phage, with which he created the first recombinant DNA molecule, and he also received Nobel Prize for that discovery. And we'll also look into the other means scientists. So first one was Herbert Boyer. So he was the co-founder of Genentech and pioneer in the recombinant DNA technology. Then comes Stanley Cohen. So he collaborated with Boyer to develop a method to clone the recombinant DNA into bacteria. And then Werner Arbor, so he discovered restriction enzymes, which made the recombinant DNA technology possible, but he himself did not create the recombinant DNA. So right answer is option B. Option C, Paulberg. Okay, so next question is type 2 restriction enzyme HIND2 was derived from option A E. coli, option B Haemophilus influenzae, option C Salmonella, and option D is Bacillus subtilis. So this is straightforward only. Yeah. So from the name only it's clear that the organism from which it is derived is Haemophilus influenzae, and this is the recognition site of HIND2, and it makes the blunt end. Okay, so yeah, for other organisms, E. coli, the enzyme which is taken is EcoR1. From Salmonella, the enzyme name is Sal1, and from Bacillus subtilis, the enzyme name is BSUR1. Yeah, and then I have kept the difference between HIND2 and HIND3 because currently, if you search for the HIND restriction enzyme, then it only returns HIND3 and not HIND2. So there is a little difference between both of them. First difference is the recognition sequence. So this is the recognition sequence of HIND2, whereas this is the recognition sequence of HIND3. So first of all, it means this upward arrow. So this means where it cuts, because the main work of restriction enzyme is to cut at the recognition site. So in this recognition site, the cut is made at the exact center, so due to which it generates a blunt end. That means on both the sides it is balanced, there is no overhang present after the cut, whereas in this case it cuts here. That means after the cut there will be overhangs which will be present in that strand. In one side there is one, and the other side there are 5 nucleotides which are hanging. Okay, so that's why it generates blunt and it generates sticky ends, or which are also known as cohesive ends. And then if we talk about discovery, then it was discovered earlier. This was the first type 2 enzyme, and this one was discovered later on, and this one is widely used in the cloning processes. Okay, so applications are like this has historical significance, but it is not used currently, and this one is the most commonly used for the molecular cloning and recombinant DNA. Okay, so next question is type 2 restriction enzyme has option A 3 subunits, option B 2 identical subunits, option C 2 different subunits, and option D is 1 subunit. Okay, so now let's see the right answer. So right answer is option B, that is 2 identical subunits. And then I have also kept the other restriction enzyme so that if question comes, then the type 1 is the multimer that contains heterotrimer. That means all the 3 are different. Type 2 is the homodimer. That means 2 similar subunits are present. So as the name suggests, 2 identical subunits, that is homodimer. Type 3 is also homodimer, and type 4 is not known. Then we talk about the recognition site pattern. So the type 1, there are 2 sites in any orientation. For type 2 it is small, generally 4 to 8 base pair long, and it is usually palindrome. That means if it is read from forward or backward, it reads out to be same. In type 3 there are 2 sites in head to head orientation and non palindromic, and these are the cleavage sites. So the cleavage site is the first or the foremost criteria with which we have divided these restriction enzyme into different types. So in type 1 restriction enzyme, it cuts the variable distance from the recognition site non-specific cleavage. That means it recognizes some sequence, but where it will cleave, that is not clear. It is variable and it is away from that site. If we talk about type 2 restriction enzymes, then the cleavage is within the type 2p or outside the type 2s, the recognition site. So type 2 are the one which can generate cohesive ends or the blunt ends, and the cleavage will be within that site. Whereas if we talk about type 3 restriction enzyme, the cleavage of one strand nicking activity is 24 to 25 base pairs from the recognition site. So in the case of type 3, if your recognition site lies here, then it will cleave 25 base pair downstream or maybe upstream. So in that way, the cleavage is not at that particular site, but it is away from the recognition site. And type 4 is like methylated only. That is the one where it cleaves. And these are the cofactors which helps this enzyme to work better. So in the type 1, the cofactors are ATP, magnesium 2 positive and SAM. In case of type 2, it is magnesium 2 positive ion, and in type 3, ATP, Mg2 positive and SAM. And in case of type 4, ATP and GTP are the cofactors. Okay, so the next question is M13 vector contains option A double stranded filamentous DNA, option B single stranded filamentous DNA, option C single stranded filamentous RNA, and option D is double stranded circular RNA. So, any guesses for this question? Yeah, you're right. So, the right answer is option B, that is single stranded filamentous DNA. So, this is the structure how it looks, and this is the single stranded DNA which is present in this M13 vector, and this is how we draw the structure when we show the different vectors, plasmids, M13, and these are the sites which are present in these vectors. And the purpose of these vectors is like, because these vectors act as a vehicle in which you can place your DNA of interest, and this vector should have certain properties. So first and foremost property is that it should have the restriction enzyme sites where it could be from where we can break it down and then insert the DNA of interest, and then it should also have ori site due to which it can replicate on its own. So this is the origin of replication site, so that it can make more copies of itself. And then in this they have shown that this is the replication endonuclease site. And there are multiple such groups and sites which are present in the M13 phage, and we'll not go into the detail, but these are the sites which are mainly either involved in the development of the capsid protein for this particular virus, or for the single strand DNA binding, then absorption proteins, minor capsid morphogenesis. So means this virus also requires some proteins, so that also are present in this particular uh, this chromosome. Okay, next question is crytoxin is produced by option A Bacillus thuringiensis, option B Bacillus subtilis, option C is P. aeruginosa, and option D is E. coli. Yeah, so you mentioned option A, yes, Bacillus thuringiensis. So it is the organism which produces this crytoxin, and this is the structure of Bacillus thuringiensis. It contains endospores, DNA, and all these. So this is the Bt toxin crystal, which Bt means Bacillus thuringiensis and toxin crystal. So whenever the plant with which we have inserted this particular gene there in the plant, which contain this toxin crystal gene, so what will happen is that as soon as this particular ball worm or other worms which eat those plants, they ingest that. So with that particular uh, means as soon as they bite this particular leaf, so there are some things which go inside. So first one is Bacillus thuringiensis, the organism. It can also go, and then it contains the this delta endotoxins and the spores which are present. So these when it ingest, then there is the means pH which is present in the gut, so that with the help of that particular pH, this particular uh, endotoxin, it gets regulated. It converts into the protoxin in its gut, and after that protoxin, with the help of proteases enzymes which are present, it converts to activated toxin, because this particular signaling molecule or other molecule get attached and it becomes activated toxin. And then this particular toxin, it interferes with the uh, membrane permeability of this particular organism, due to which the balance will be disturbed and the insect will die. So that is how the bt crops, which is means which was very well known. So bt crops means because they contain the gene which give rise to this particular toxin, so that's why the name bt toxin came from there. Okay, so next question is bt plants are engineered to control option A weeds, option B lepidopteran pests, option C fungi, and option D is bacteria. Yeah, so right answer is option B, which is lepidopteran pests. because weeds are the other plants which interfere with the growth of your plant, and fungi and bacteria are also not means involved in this particular thing. So these are the lepidopteran pests which are generally controlled with the help of these bt plants. So next question is oric is the site of option A transcription, option B translation, option C replication, and option D is cell division. Yeah, so you're right, oric is for the replication, and its full form is also even origin of replication. And this is the site which must be present in your vector so that it can divide on its own, it can replicate. Yeah, and in normal genome also this oric site is present. Okay, so now we'll talk about the week 2 mcqs. So in week 2 you have looked into the breakage of genomic dna and its repair. So we'll discuss mcqs in the week 2. So in the week 2, first question is which one of these is not an endogenous source of DNA damage? Option A DNA base mismatch, option B DNA methylation, option C ultraviolet radiations, and option D is topoisomerase DNA complexes. Okay, so you mentioned option B. So in this case, the right answer is option C, because they have mentioned which one is not an endogenous source. That means the source which, in this you have to tell the exogenous source, means external source. Whereas these other sources, these all are endogenous sources, and ultraviolet radiation is the exogenous. And in this figure I have shown that which are the endogenous and which are the exogenous sources. So endogenous sources include reactive oxygen species or any enzymes which are involved in the DNA polymerase if there is some fault in them, and then uh, the endocrine substances which are released in the body, so they come under endogenous sources of DNA damage. Whereas in exogenous sources, chemotherapy, uv radiations, infrared radiations, and viruses which are external sources which leads to DNA damage. And DNA damage can be due to base mismatches, single strand breaks, double strand breaks, interstrand crosslinks, DNA adducts, and interstrand crosslinks. So in this case, right answer was option C, which is UV radiations. Okay, next question is choose the correct sentence regarding the DNA base mismatches. So option A is DNA mismatches are the defects in DNA that occur due to the complementary base alignment in the same base pair of the DNA duplex. Option B was a transition is when a purine nucleotide is changed into pyrimidine or a pyrimidine nucleotide is changed into purine. Option C, transversions are any mutations in which a purine is replaced with a pyrimidine or vice versa. And option D is transitions are more likely to change the amino acid sequence of proteins than transversions. So you need to tell the correct sentence regarding the DNA base mismatches. Okay, so let's look at the right answer. So the right answer is option C, which means transversions are any mutations in which purine is replaced by the pyrimidine or vice versa. So in this case, the definition was opposite. So transition means when the pyrimidines are replaced with pyrimidines and purines are replaced with purines. And this is what it shows. So from G to A, that means both are purines. And if A mutates to G, then it is transition type of mutation. If G converts to T, that is thymine and cytosine, they are pyrimidines. So when guanine changed to pyrimidine, that is thymine, so it is transversion. So keep this thing in mind that these type of mutations where these nitrogenous bases converts from particular type to other type, that are transversions, and if they mutate within their own type, then they are transitions. Okay, so next question is deamination is the process of removing dash from a molecule. Option A, an amino group, option B, a carboxyl group, option C, hydrogen, and option D is none of the above. Yeah, so both are right, the amino group, and it is also evident from its name, deamination, that means removing the amino group. And this is how, if we remove the amino group from cytosine, then it converts into the uracil. And similarly, if we convert 5-methyl cytosine, because there are methylations and all the things are going on inside our body, so whenever cytosine get methylated, and its deamination leads to the thiamine formation. Okay, so these type of mutations, these type of DNA errors, damages are, means deamination processes, so they will come under the endogenous source of DNA damage. Okay, next question is, which one of these is not true regarding the alkylating agents? Option A, alkylating agents transfer the alkyl groups to the DNA. Option B, simple methylating agents generate adducts at the hydrogen and carbon atoms in the DNA bases. Option C, O alkylations are strong mutagens, whereas N alkylations are less mutagenic. And option D is N alkylated purines are the primary product of DNA methylation. Okay, so I mentioned option C. Uh, yeah, so one more point which I want to make here is that uh, when we were discussing this question during our week 2 session, so that time uh, I mentioned option C only. So that option C is marked as correct according to the previous assignments which were solved for this particular course. But then I searched the internet and I got to know that the right answer is option D. So in the this question they have asked that the statement which is not true regarding the alkylating agents, and in this it says that N-alkylated purines are the primary product of DNA methylation. So in this case these are frequently formed, but these are not the primary products, and the most mutagenic lessons they generally arise from O alkylation. So this statement is incorrect, and as at the same time the C statement, this is also somewhat correct only. So it says that O alkylations are the strong mutagens. So this is true that they are highly mutagenic, and it says that whereas N alkylations are less mutagenic. So N alkylation is often causes less severe lessons, but we cannot say that it is less mutagenic. So in this case there is still a confusion. So if whether you take C option or D option, it will be right. And if we say option A, that is alkylating agents transfer alkyl groups to DNA, so this is right because it transfers, so this is true. And then option B was simple methylating agents generate adducts at the hydrogen and carbon atoms in the DNA bases, so this is also true. Whereas these two options, so there is little confusion between them because they appear to be correct only, so any option you can select. Okay, so let's move to next question. Next question is, name the process by which UV inactivated organisms restore their function by repairing UV induced DNA damages with the energy of near UV light. Option A, base excision repair. Option B, mismatch repair. Option C, translation synthesis. And option D is photoreactivation. Yeah, so you mentioned option D, which is photoreactivation. So this is right, and this is the photoreactivation repair. So generally if there are pyrimidine dimers, then with the help of exposure of UV radiations or visible light, so it get repaired, and the enzyme photolyase that is responsible for this conversion. So and this type of repairing process is known as photoreactivation. Okay, so next question is, name the enzyme involved in base excision repair. Option A, DNA glycosylase. Option B, DNA polymerase. Option C is DNA topoisomerase. And option D is RNA polymerase. So base excision repair. Okay, so in this case the right answer was option A, which is DNA glycosylase, and this is the entire base excision repair process. So base excision repair is generally used when if there is only a single base, if there is problem with just one base, so then base excision repair mechanism get activated. So in this case this is the deaminated C. So due to deamination of cytosine, it got converted into the uracil. So we have previously seen that how it converts. So now in one of my strand, double strand, so one of my strand, it contains U in place of C. So when this particular strand get replicated, so it will have the complementary strand which will contain A. So due to which the information got mutated, it got changed. So to repair this type of damage, firstly, uracil DNA glycosylase will come and it will remove the unwanted base uracil. So glycosylase work is to remove that particular base which is faulty. So it removes that base and it will generate a site which will be AP site. So depending on the base, it could either be apyrimidinic site or apurinic site. That means the site which lacks the purine or pyrimidine. And after that, AP endonucleases and phosphodiesterases will come, which will break the phosphodiester bond and will create a gap here. And then they will cleave this AP site, and the gap will be there. So then later on, DNA polymerases and ligases will come, which will add the new DNA and will seal the gap. So with the help of ligase, the gap will be sealed, and with the help of DNA polymerase, the complementary nitrogenous base will come. So complementary DNTP will come and it will bind here. So that is how this damage is repaired. And in this case, if we talk about DNA polymerase, so you all know that DNA polymerase work is just of the extension that it will bring the complementary DNTP and will join it. DNA ligase will seal the gap due to those broken phosphodiester bonds will be then sealed, they will be generated. So that is the work of DNA ligase. RNA polymerase work is to synthesize the RNA strand, so it is not used here. And DNA topoisomerase, it is also known as DNA gyrase, and its work is to relax the relax the supercoiling which takes place during the unwinding of DNA. So in case of base excision repair, the topoisomerase is generally not used because in this case we are not unwinding the DNA as it is unwind in the case of replication. So there is no supercoiling which is taking place. So that's why it's not the right answer. Okay, next question is, which of these is not involved in single strand break repair? Option A, PARP1, which is poly-ADP ribose polymerase 1. Option B, XRCC1, which is X-ray repair cross complementing protein 1. Option C is TLS polymerase, and option D is PNKP, which is polynucleotide kinase 3 phosphate. So which one is not involved in single strand break repair? Yeah, you both are right. So right answer is option C, which is TLS polymerase. So it is not involved, whereas PARP1, XRCC1 and PNKP are involved. And in this I have shown all these enzymes where they are used. So first of all, this is the topoisomerase 1 DNA link, and this one is endogenous reactive oxygen species hydrogen peroxide. So these are the sources of this particular single strand break. And then SSP detection. So for the detection of the single strand break, this PARP1, PARG, these enzymes come and they detect where the single strand break has taken place. And after, as soon as they identify the site, then the next process is DNA end processing. So for this DNA end processing, XRCC, APTX, PNKP, POL-B, polymerase beta, so these all come, and whereas these TDP1, LIG3, XRCC1, PNKP, so they all come here depending on what kind of factor has caused that damage, and later on one of them they leads to DNA gap filling and DNA ligation. So that is how this single strand break repair takes place. Next question is, which of these disorders is associated with severe photosensitivity resulting in sunburn and skin pigmentary changes? Option A, Fanconi anemia. Option B, xeroderma pigmentosum. Option C, cocaine syndrome. And option D is trichothiodystrophy. So it's related to photosensitivity results. Photosensitivity. Yeah, so you're right. Option B is the right answer, which is xeroderma pigmentosa. And this generally happens when you are exposed to too much UV radiations or light. So in this case, the most kind of damage which is mostly discussed in xeroderma pigmentosa is the thiamine dimers. And this is how the patients look, and they are very sensitive towards the light. So whenever they goes in the sunlight, they develop sunburns, skin pigmentary changes. Okay, so I'll not go into this detail, but generally it is discussed that how this what happens with the xeroderma pigmentosa skin versus the normal skin. So next question is, choose the correct group of proteins involved in E. coli DNA mismatch repair pathway. Option A, MUT-S, MUT-L, MUT-H, DNA helicase 2, exonuclease, DNA polymerase 3 holoenzyme, and DNA ligase. So you have matched the correct order, that from where which enzymes come first and which comes afterwards. And option B is mut S, mut L, mut H, DNA helicase 2, exonuclease, DNA polymerase 3 holoenzyme, and RNA primase. Option C, mut S, mut L, mut H, DNA helicase 2, exonuclease, RNA polymerase 3 holoenzyme, and DNA ligase. And option D is mut S, mut L, mut H, DNA helicase 2, endonuclease, DNA polymerase 3 holoenzyme, and DNA ligase. Sorry, so now you know what is the right answer. So the right answer is option A, which is mut S, mut L, mut H. Then comes DNA helicase 2, then exonuclease, DNA polymerase 3, and DNA ligase. So in first of all, we can cancel this out because RNA primase is not there. Then we can cancel this option also out because it says RNA polymerase, so this is also not right. Whereas in this case it says endonuclease, so in this case the nuclease one is the exonucleases. So that is how option A is correct, and this shows that if in mismatch repair generally there is mismatch in the bases present on both the strands. So here it is G and here it's T, so this is mismatch. Now mut S, mut L, and mut H comes first, and they recognize that where is that mismatch present, and then later on, after the identification of mismatch, DNA helicase, single strand binding proteins, ATP, all of them will come, and these helicases will start the unwinding of this particular DNA. And after that, exonucleases will come, RECJ, exonucleases 7, exo 1, exo X, all of them will come and they will cleave this one of the strand of the DNA where the mismatch was present. So there are some other uh, enzymes also which detect. So generally the strand, which one is the parent strand or the original strand, it is generally methylated, so that we can distinguish that which strand is the older strand, and means which strand should we cleave. So this parent strand which was methylated, it is not cleaved, but the other strand is generally cleaved, so that mismatch could be removed. And after cleaving this, the DNA polymerase will come and DNA ligase will come, which will synthesize the correct strand, which will correct this mismatch, and it will synthesize the nucleotides which are complementary to the parent strand. And later on, ligase will seal the gaps, it will create the phosphodiester bonds. So this is the order of enzymes which are used here. Okay, so next question is, which one of these is the enzyme which is involved in repairing the DNA damage which is caused by the caused by exposure to the UV light? So option A is DNA glycosylase, option B is DNA helicase, option C is DNA ligase, and option D is DNA photolyase. Yeah, so you're right. So answer is option D, which is DNA photolyase. And this is what it shows, that this is the intact um, means DNA strand in which two nucleotides which are along with all the groups. So these are intact nucleotides present in the DNA strand, and this is this means exposure to the sunlight. So if there are UV lights, then what will happen? This intact uh, nitrogenous bases which are present in the DNA, so they will make the double strand. They will make the means they will bind to one another. So there will be we can say it as the intra-strand bonds will take place, due to which thymine dimers will form. And then this is known as the damaged DNA strand, because in this thymine dimer has formed. So with the help of photolyase enzyme, it will cleave that thymine dimers, it will break that intra-strand bonds, and it will again convert it into the intact nucleotides which are present in the double strand. So in this case it's not much clear, but the basic idea is that the photolyase is the enzyme which will means treat that particular damage which takes place due to the presence of the UV light. Okay, so next question is, which DNA repair pathway is primarily responsible for repairing the bulky adducts such as thymine dimers? Option A is base excision repair, option B is nucleotide excision repair, option C is mismatch repair, and option D is non homologous end joining. Okay, so the right answer is option B, which is nucleotide excision repair. And the main difference between the base excision and nucleotide excision is that only that in base excision only one base is faulty. If more than one base is faulty, then we use the nucleotide excision repair, in which longer fragment gets deleted, means it gets removed, and then we synthesize the fresh uh, strand or fresh group of nucleotides against that particular parent strand. Okay, so next question is which protein complex initiates the global genome nucleotide excision repair, which is also known as GGNER? Option A, RNA polymerase 2. Option B, XPC HR HHR23B. Option C, MUT-S, Option D is PARP1. Yeah, so the right answer is option B, which is XPC and HHR23B. So this nucleotide excision repair, it can be divided into two types. One is transcription coupled repair, in which transcription is also taking place at the same time, and other one is global excision repair. So in the case of global excision repair, the enzymes or the protein complexes were the XPC and then HHR23B, which takes place here, and then damage is recognized, and all the other protein complexes come in, and then it will remove that particular damage, and it will repair that damage. Whereas if MUT-S, MUT-L, these are used in the mismatch repair, and RNA polymerase 3 is not involved in DNA repair because this is for the synthesis of RNA. So this is not right. And similarly, this one also we have discussed that this was used previously. This is not used in the global excision repair, and this do not initiates this particular process. Okay, next question is in transcription coupled NER, what triggers the recognition of DNA damage? Option A, DDB2 binding. Option B, RAD51 recruitment. Option C, RNA polymerase stalling. And option D is DNA ligase activity. Yeah, so you both are right. So RNA polymerase stalling is the phenomena which triggers the recognition of DNA damage, because RNA polymerase's work is to synthesize the RNA strand, which will be the new strand which will be synthesized against the DNA template. So whenever the polymerase gets stalled, that means there is some damage in the DNA. So that is how the initiation is done, and that is what is mentioned here. RNA polymerase stalls. Okay, next question is what type of mutation results when the 5-methyl cytosine undergo deamination? Option A, C2A transversion. Option B, G2A transition. Option C, C2T transition. And option D is G2T transversion. Okay, so let's see. So the right answer is option C, which is C2T transition. So 5-methyl cytosine, it gets deaminated and get converted into thymine. So this is the transition because both of them are pyrimidines. Okay, next question is which of the following is a bifunctional DNA glycosylase? Option A, SMUG1, option B, NILE1, option C, APE1, and option D is XRCC1. So which is bifunctional DNA glycosylase, and all of them are glycosylases only. Yeah, you're right. So right answer is option B, which is NILE1. This is bifunctional DNA glycosylase, and why it is bifunctional? Because it has both the glycosylase and AP ligase activity, which removes the damaged bases and cleave the DNA backbone. That's why it is bifunctional. Whereas all others, SMUG1 is the monofunctional DNA glycosylase, and it only removes 8-oxoguanine, and it does not have the ligase activity. Whereas APE1 is the AP endonuclease, so it is not a glycosylase, and XRCC1 is also the one which is involved in the single strand break repair and not a glycosylase. Okay, so in this I have kept the example of monofunctional and bifunctional glycosylases. So these are the examples, means just in case if they come in exam, but this is not much important with the examination perspective. But if you're interested, you can see that OGG1, NTH1, NILE1, NILE2, NILE3, these all are the bifunctional AP glycos and DNA glycosylases. and these are the monofunctional ones. Okay, so next question is, which DNA repair mechanism uses the polymerase NETA to bypass the UV induced thymine dimers? Option A, base excision repair. Option B, homologous recombination. Option C, translation synthesis. And option D is nucleotide excision repair. Yeah, so the right answer is option C. Yeah, you both are right, which is translation synthesis, because that only use polymerase NETA. Otherwise, rest others use DNA polymerase 3 or other type of polymerases or polymerase 1. But in this case, polymerase NETA is used. Translation synthesis, because in this case, the translation as a structure, lesson like structure is formed. And then this is also known as the SOS repair in the E. coli and in humans also. Okay, so in this case, it's shown that it bypass the lesson wherever the DNA lesson is formed. That is a special structure of the DNA which is faulty, which leads to the DNA damage. So it leads to the extension, and then other polymerase kappa will come, and then it will synthesize that strand where the damage has happened, where the lesson is present. So that is not much important. The main thing is that this particular polymerase NETA is associated with translation synthesis, and if you are interested you can read it in detail. But the main thing is that this is also known as SOS repair, which is save our soul kind of repair. This only gets activated if all other repair mechanisms get failed, and this is also error prone repair. So if organism has no other repair mechanisms working, then only this will get activated to just save it, but this can have many errors as well. So next question is, Fanconi anemia is primarily caused by the defects in the repair of option A, double strand breaks. Option B, oxidative base damage. Option C is interstrand crosslinks. And option D is mismatch base pairs. Okay, so this I'll tell. So the right answer is option C, which is interstrand crosslinks. Okay, so interstrand means between two strands. So in this case, the link which is formed between the two different strands, and this link is not normal because it is taking place between two different nitrogenous bases which are present away from each other. So this type of defect is known as interstrand crosslink, and this is observed in the Fanconi anemia. If that particular cross-linking or the bond is present between the same nitrogenous bases present on the same strand, then it is intra-strand crosslink, which we see in the case of thymine dimers. This is the DNA protein crosslink, and this is the monoadduct in which something is attached to one of the nitrogenous base, which could either be alkyl group or other groups. Yeah, so the right answer in this case was interstrand crosslinks. Okay, so next question is, the main function of the protein PARP1 in DNA repair is to option A, join the Okazaki fragments. Option B, recruit repair proteins to single strand breaks. Option C, excise damaged bases. And option D is remove the thymine dimers. So this is the PARP1. Yeah, so you're right. Right answer is option B, which is to recruit the repair proteins to the single strand break site. Yeah, and generally this particular enzyme gets activated if there is the alkylating agents or oxidative stress present, and it also activates the replication, SSB repair, double strand break repair, DNA degradation, which leads to these types of processes leads to cell survival, and DNA degradation leads to cell death. So ultimately it maintains the stability of the genome. Okay, next question is, which enzyme removes the 3' phosphate group during the single strand break repair? Option A, DNA polymerase beta. Option B, TDP1. Option C, PNKP. And option D is XPA. Yeah, so right answer is option C, which is PNKP, that is polynucleotide kinase phosphatase, because as the name suggests, phosphatase. So it has both the activity as kinase and phosphatase. With its phosphatase activity, it removes the 3' phosphate group. Okay, next question is, what is the role of TF2H in the nucleotide excision repair? Option A, cut the DNA strand near the lesson. Option B, unwind the DNA helix around the listen. Option C, fill the gap with new nucleotides. And option D is ligate the nick in the DNA. Yeah, so you're right. So the right answer is option B, which is unwind the DNA helix around the listen. That is the work of TF2H. This is the TF2H and it leads to local unwinding of the DNA. Okay, so with this, now we'll move to week 3. Okay, so I think I have kept 20 MCQs in each of the week sessions, so that is taking long time. So now we'll just quickly go through main 10 MCQs which have come in your previous week assignments and they might come in your exam. So in week 3, the topics were homologous and non-homologous recombination and site-specific recombination. So in this, the first question is, what are the two broad types of DNA damage? Option A, mechanical and chemical. Option B, endogenous and exogenous. Option C, internal and external. And option D is primary and secondary. So you need to tell the broad DNA damage types. Yes, that's right, endogenous and exogenous. And we've already discussed when we were talking about the UV radiations that they are the exogenous source of DNA damage. Okay, next question is, a Chi site is option A, replication hotspot, option B, DNA damage hotspot, option C, recombination hotspot, and option D is DNA repair hotspot. Yeah, you're right. So Chi site is the recombination hotspot. As you can see in this case, it is present here. So this is the Chi site. This is the recombination hotspot. So it's called recombination hotspot because Chi site is a specific and short DNA sequence which stimulates the homologous recombination by altering the activity of rec BCD complex. And when this rec BCD complex encounters this Chi site, it stops the degrading the 3' end and it loads the rec A and initiates the strand invasion and making the recombination much more likely at the location compared to rest of the genome. Okay, so this rec C is generally, these are involved in the recombination and this Chi site is more responsible towards that. So that site is known as recombination hotspot. Yeah. So next question is, which repair pathway is considered high fidelity? Option A, non-homologous end joining. Option B, homologous recombination. Option C, base excision repair. And option D is mismatch repair. Yeah, you're right. So the high fidelity is the homologous recombination. If we talk about NHEJ, it is low fidelity for double strand breaks because it directly ligates the DNA ends without a template, because it is not using other template, so it might insert insertions or deletions. Homologous recombination is high fidelity because it uses intact homologous DNA sequence as a template to restore the original sequence precisely. Whereas if we talk about base excision repair, so that is also high accuracy for the single base damage but for the small lessons and not large breaks. So small lesson means just one base pair. And if we talk about mismatch repair, so this is high accuracy for correcting the replication errors, but it's just specific to the mismatches and not for the double strand breaks. So that's why right answer is option B. Next question is, what binds to the DNA ends in the initial step of double strand break repair? Option A, RAD51, option B, RPA, option C, MRN or Ku7080 heterodimer, and option D is rec BCD. Okay, so you are right, option C is the right answer, which is MRN or Ku7080 heterodimer. So this is where it binds in the double strand break in the initial step. So yeah, and this is in the case of non-homologous end joining, whereas in the case of uh, homologous recombination, the MRN is the one. The MRN complex is mainly involved, which is the complex made up of MRE11, RAD50 and NBS protein complex. So that is the MRN, and Ku7080 is the non-homologous end joining. repair. Okay, next question is which component of rec BCD recognizes the chi sequence? Option A rec A, option B rec B, option C rec C, and option D is rec D. Yeah, so the rec C is the one which recognizes this chi sequence. Okay, next question is rec A catalyzes which of the following? Option A ATP driven homologous pairing and strand exchange of DNA molecules necessary for the DNA recombinational repair. Option B hydrolysis of ATP in the presence of single stranded DNA. Option C ATP dependent uptake of single stranded DNA by duplex DNA and ATP dependent hybridization of homologous single stranded DNAs. And option D is all of the above. Okay, so in this case, the right answer is option D, that is all of the above. So, all of these processes are catalyzed by rec A. Means option A is right, but option B and C are also right. So, that's why option D is the right answer. Okay, next question is in the context of site specific recombination, what is the function of tyrosine recombinase catalytic mechanism? Option A introduction of double strand breaks and rotation of cleaved DNA segments. Option B formation of holiday junction intermediate and resolution. Option C degradation of mismatched DNA segments. And option D is extension of DNA by adding the nucleotides. Okay, so you mentioned option A. So in this case, you mentioned option B. So in this case, the right answer is option B, which is the formation of holiday junction intermediate and resolution. So in the case of tyrosine recombinase, there is the formation of holiday junction. And in this case, where they mentioned that introduction of double strand break and rotation, so rotation takes place in the case of serine recombinases. So this is the work of serine recombinases. Holiday junction is observed in the case of tyrosine recombinase. Okay, so next question is which site specific recombinase is known for its use in genetic engineering to induce the recombination at specific sites in the genome? Option A Cre recombinase, option B Rec A, option C Roof C, and option D is Rec G. Yeah, so you're right. The Cre recombinase is the one which is site specific recombinase, and whenever we talk about the Cre LOXP mouse, so this is the recombinase which is generally involved. Okay. So next question is which protein is essential for the excision of the phage DNA from bacterial DNA in the phage lambda phage system? Option A int protein, option B XIS protein, option C both int and XIS proteins, and option D is Cre recombinase. So you have to tell the protein essential for the excision. Okay, so the right answer is option C. That means both int and XIS both are involved. Okay, so for integration int and for excision XIS. So excision int is also used in that. So that's why both int and XIS. And here it is mentioned that bacterial chromosome requires both int and XIS working together. Int performs the strand cleavage and rejoining, whereas XIS facilitates the site alignment and directionality towards the excision. So although in integration only int is involved, but in excision both int and XIS proteins are involved. So keep that thing in mind. Next question is what triggers the formation of tetrameric species in syn resolvase, which is also known as serine resolvase? So option E is DNA polymerase activity. Option B accessory protein high local concentration or mutations. Option C RNA interference, and option D is spontaneous DNA damage. Yeah, so you're right. Option B is the right answer. That is accessory proteins, high local concentration and mutations. They triggers the formation of tetrameric species. Yeah, and that is all for this. And this is how the tetrameric complex is formed. Yeah, and in this case the rotation is involved, which you were talking previously. So in this case, attachment, cleavage, attachment, and then subunit rotation, and then religation and they further dissociate. So these are the dimeric units and they combine and make a tetrameric unit. Okay, so now we'll directly jump to the next week because otherwise it will take a lot of time. So now we'll jump to week 4, in which we'll look into the targeted genetic modifications. So first question is, which of the following function does Cre recombinase exhibit? Option A, excision, option B, inversion, option C, translocation, and option D is all the above. Okay, so in this case the right answer is option D, that is all the above. So in this I have shown that where the Cre recombinase is used. So it depends that in which direction the LOXP site orients, that in what is the direction of LOXP site, and direction, with that direction I mean when we read that sequence. So where from which direction it can be read. So in this case, if both of them facing towards each other, then in that case, whenever the Cre recombinase will perform recombination, so it will lead to the inversion of the gene which is present in between that site. And if both the LOXP sites are directing towards the same direction, in that case there is deletion of the gene. So in this case, one LOXP site has the genome, whereas other segment, that particular gene segment is removed from the entire genome, and it is means circularized in this case, and one LOXP site is also present with this gene X. And if these LOXP sites, if they are present on two different different strands of the DNA, then it leads to the translocation. This Cre recombinase will lead to the translocation, so this particular segment get exchanged. So means the segment exchange takes place in this case between the LOXP site. So this transfers here and this comes down. So that is translocation. So that is how the Cre recombinase is involved both in all these processes: inversion, deletion, and translocation. Next question is, flippase, that is FLP recombinase, is derived from which of the following organisms? Option A, Escherichia coli, option B, Haemophilus influenzae, option C, Saccharomyces cerevisiae, and option D is Bacteriophage P1. Yeah, so you're right. It is obtained from Saccharomyces cerevisiae, which is known as yeast. So next question is, Dash devised the technique of introducing DNA directly into the nucleus of the cell using the microinjection. Option A, Michael Wigler, option B, Martin J. Evans, option C, Oliver Smithis, and option D is Mario Capecchi. So you need to tell the one of them who has invented this microinjection for this direct delivery. Yeah, so you're right. Dr. Mario Capecchi was the one who introduced this technique. So next question is, tamoxifen dependent Cre recombinase contains the ligand binding domain of which of the following receptors? Option A, estrogen receptor, option B, retinoid receptor, option C, thyroid hormone receptor, and option D is glucocorticoid receptors. Okay, so now let's see. So right answer is option A, that is estrogen receptor. So when we talk about the tamoxifen dependent Cre recombinase, so tamoxifen is this particular chemical. So this is inactive, and this particular tamoxifen, when it enters into the cell, then it binds to the ER, that is estrogen receptor. So this is the site where the tamoxifen binds. So that contains the ligand binding site. And that is how with the binding of this it can enter into the nucleus, and then it can activate the Cre recombinase to bring about the recombination process. Okay, next question is, with respect to TET-on, that is RTTA system, which of the following statement is true? Option A, Cre recombinase is expressed only in the presence of tetracycline and doxycycline. Option B, Cre recombinase is expressed only in the absence of tetracycline or doxycycline. Option C, tetracycline does not affect the expression of Cre recombinase. And option D is, presence of tetracycline prevents the binding of TTA to TET responsive promoters. Okay, so you mentioned option A, and that is right. So Cre recombinase is expressed only in the presence of tetracycline and doxycycline, and that is also clear from its name, TET-on. So TET-on means when tetracycline is present, it is getting expressed. Tet off means if tetracycline is present, then it is off. So that is the main thing between tet off and tet on. And in this system, the tetracycline responsive promoter is present, and it is expressed only when this tetracycline and doxycycline are present. Okay, next question is, what purpose does the introduction of HSV thymidine kinase gene into the vector serve? Option A, act as a positive selection marker. Option B, protects the target gene from disintegration within the host cell. Option C, act as a negative selection marker. And option D is helps in the excision of the target DNA within the host cell. So HSV thymidine kinase. Yeah, so you mentioned option C, and that's right. So it acts as a negative selection marker. Okay, so this HSV T-gate phosphorylates the Gen-C clovir, which is FIAU. It produces the toxic nucleotides that will kill the cell. That's why it acts like a negative selection marker. Okay, so next question is, which is the most commonly used antibiotic in the selection of cells with the targeted gene knockout? Ampicillin, neomycin, streptomycin, or penicillin? Okay, so you mentioned option A, but the right answer is option B, that is neomycin. So ampicillin is used for the bacterial selection, and it is not used for the this ES cells, and mostly means mammalian ES cells this ampicillin is not used, whereas in the case of neomycin this is widely used in the mammalian cell selection. NeoR, which is the neo-resistance marker gene, it is generally used in the knockout constructs. And if we talk about streptomycin, so it is common in bacterial culture, but it is not used for the knockout studies. And similarly, penicillin also, so antibiotic for the bacterial contamination control, but not for the gene targeting in this targeted gene knockout. Okay, next question is, embryonic stem cells are derived from which part of the blastocyst? Option A, trophoblast. Option B, blastocele. Option C, zona pellucida. And option D is inner cell mass. Yeah, so you're right. Option D is the one. So it is obtained from the inner cell mass. And these embryonic stem cells, these are totipotent in nature. That means they can give rise to any cells in the body. And later on, mesoderm, ectoderm, endoderm. So these are the pluripotent cells which can further give rise to one particular category of cells. Okay, so next question is, what is the restriction enzyme site located within the homology arm of an insertion type vector is used for? Option A, to linearize the vector construct for homologous recombination to occur. Option B, to integrate the gene of interest into the vector. Option C, to excise out the gene of interest from the vector. And option D, to integrate the negative selection marker into the vector. So just keep this thing in mind that they have talked about homology arm, and then restriction enzyme site present in that particular homology arm. So what is the purpose? Yeah, so option A is right, to linearize the vector construct for the homologous recombination to occur. Okay, so the next question is, which strain of mice is most commonly used for constructing the targeting vectors? Option A, C57BL by 6. Option B, BALB by C. And option C is 129 by SV. And option D is FVB by N. Okay, so you are right. Option C is the right answer. That is 129 by SV. And this is the standard strain for the ES cell derivation, embryonic stem cell derivation, and most targeting vectors are constructed using the DNA from this particular strain, whereas this C57BL6, this is common as the blastocyst recipients, but it is not the main source of ES cells in targeting vectors. So these are other strains of this particular mice, but these are not generally used, and this one is the strain which is used. Okay, now we'll skip other questions and let's move to week 5. So in week 5 we looked into zinc finger nuclease technology for the genome editing. So in this case, first question is, which of the following statements accurately describes the alpha helix secondary structure in proteins? So option A is the alpha helix is stabilized by the hydrogen bonds between the side chains of amino acids. Option B, each turn of alpha helix contains 5.4 amino acid residues and has a vertical distance of 1.5 angstrom between the equivalent atoms. Option C, the alpha helix has 3.6 amino acid residues per turn with a vertical distance of 5.4 angstrom between the structurally equivalent atoms in a turn. And option D is the alpha helix is the least common secondary structure and is stabilized by the ionic bonds. Okay, so I mentioned option C and that is right. So this is the structure of alpha helix in which there are 3.6 amino acids or amino acid residues per turn, and each amino acid rotates the helix at 100 degrees. This is the typical length. So this is the alpha helix, and then there are other categories of uh, alpha helix, 310 helix, and then other helix. So in this case, rise is 1.5 angstrom, that is the distance between the two residues, which is the distance. So in the helix, after the one turn, so that is 5.4 angstrom. One turn is of nearly 5.4 angstrom, and then the hydrogen bonding takes place in this secondary structure, the alpha helical structure. So the in secondary structures of the proteins, the major bonds are the hydrogen bonds which takes place, because in this it is mentioned ionic bonds. So ionic bonds are not involved in the secondary structures, they come when we talk about the tertiary structures. And in this case, in alpha helix, the hydrogen bond is present between the first residue and then between the I plus fourth residue. So first and fifth residue there will be a hydrogen bond. Okay, so next question is, which of the following best describe the function of fork1 restriction endonuclease? So option A, fork1 recognizes symmetric nucleotide sequences and introduces blunt end cuts directly at the recognition site. Option B, fork1 belongs to the class of restriction endonuclease that recognizes the specific but asymmetric nucleotide sequences and introduce the staggered cleavage at the positions away from the recognition sequences. Option C, fork1 cleaves DNA randomly without recognizing the specific sequence and creates the blunt ends. Option D, fork1 is a restriction endonuclease that recognizes palindromic sequences and make the cuts at the recognition site, resulting in the blunt ends. Okay, so you have mentioned option D. So right answer is option B, that is the fork1 belongs to the restriction endonucleases that recognize specific but asymmetric nucleotide sequence and introduced a staggered cleavage at positions away from the recognition sequences. So one takeaway from here is that as it is fork1. So first of all, fork1 means this is one means this is the type 1 restriction endonuclease. So when we were talking about the difference between different types, then we studied that type 1 restriction enzyme is the type of restriction endonucleases which cleaves the means downstream of the recognition site, away from the recognition site. That can be anywhere. And then the sequence which they recognize is the asymmetric nucleotide sequence that do not have any symmetry. So that's why it is asymmetric in nature, and it introduces the staggered cut. So staggered cut we mean sticky end kind of cut, not blunt. So that's why the option B was right. Okay, so next question is, which of the following features are characteristic of the C2H2 zinc finger motif? Option A, it contains a repeated 20 to 25 amino acid sequence with a single conserved histidine and no cysteines. Option B, it is primarily found in RNA binding proteins and does not interact with DNA. Option C, it includes 25 to 30 amino acid sequence with 2 conserved cysteines and 2 conserved histidines and is involved in transcription regulation and protein-protein interactions. And option D, it consists of single zinc finger motif and functions solely in lipid binding. Yeah, so you mentioned option C, that is right. It includes 25 to 30 amino acid sequences, and its name C2H2 only suggests C means cysteine and H means histidine. So C2H2 means there are 2 cysteines and 2 histidines which are conserved and generally involved in the transcription regulation and protein-protein interactions. And then there is a zinc with which these two cysteine and 2 hydrogens they coordinate with that zinc, and this is the structure which it adopts. So there are multiple such motifs. These are 3 motifs, and then domain contains multiple motifs. So this is a zinc finger domain which has 3 motifs. Okay. Next question is, what is a key characteristic of modular assembly method for zinc finger protein design? Option A, it uses a single zinc finger to target a 9 to 12 base pair sequence. Option B, individual zinc fingers are optimized against the target triplet DNA sequences and linked together to form the 3 or 4 ZFP targeting 9 or 12 base pair sequences. Option C, it involves a context dependent assembly of zinc finger arrays without optimizing individual zinc fingers. And option D, it employs a high throughput approach to design the zinc finger proteins targeting the long DNA sequences. Yeah, so you're right. Option B is the right answer. That is, individual zinc fingers are optimized against the targeted triplet DNA sequences. Because we know each of these unit will bind to 3 nucleotides, that is triplet of the DNA sequence. So modular assembly means we can selectively join these different motifs which are selective towards these 3. So for example, if I have a DNA site where I want my this ZFN to bind, because in this particular protein we want it to bind to that particular site so that it can bring about the cleavage at that site. So for editing we use it. So for that reason we can modularly design it. We can join that specific zinc finger domains which are specific to these triplet of the nucleotides. So that is the advantage, and these target the DNA triplets and these are linked together to form 3 4 ZFPs which will target 9 or 12 base pair sequences. So if there are 3 ZFPs, so 3 will target 9, means 9 will be the length of the sequence which it will target, and if there are 4 such motifs which are used, then it will target 12 nitrogenous bases or 12 base pair sequence. Okay, next question is, which of the following steps is not involved in ZFN mediated gene targeting? Option A, identifying ZFN target sites near the targeted locus within the gene X of interest. Option B, designing and selecting the zinc finger proteins that recognize the chosen ZFN target sites. Option C, converting the designed and selected ZFPs into the zinc finger nucleases. And option D, applying a high throughput screening method to test multiple gene editing outcomes simultaneously. Okay, so option D is right, which is all other options are true related to ZFN mediated gene targeting, whereas this is the step which is not involved. Okay, next question is, which of the following is a key factor for successful direct delivery of ZFN proteins into the cells? So option A is low lipophilicity and high pH sensitivity of ZFN proteins, which require the modification to enhance the cell membrane penetration. Option B, the positive charge of cysteine to histidine 2 zinc finger domains in ZFN proteins, which aids in penetrating the cell membrane. Option C, high degradation rate of ZFN proteins, which necessitates the frequent redelivery to maintain the effectiveness. And option D, the use of non-enzymatic methods for protein delivery, which bypass the need for stability modifications. Okay, so you mentioned option B, and that is right. So this is the due to the positive charge of cysteine and histidine zinc finger domains, which helps in penetrating into the cell membrane. So next question is, what is the primary function of ribosomal skipping in translation? Option A, it enhances the ribosome's ability to bind to multiple mRNA molecules simultaneously. Option B, it allows for the incorporation of additional amino acids into the growing polypeptide chain. Option C, it prevents the ribosome from linking a new amino acid, leading to co-translational cleavage of polyprotein. And option D is it ensures that the ribosomes only translate viral peptides and exclude the cellular proteins. Okay, so you mentioned option C, and that's right, because it prevents the ribosome from linking a new amino acid, which will lead to the co-translational cleavage of polyprotein. Next question is, which repair process is utilized by the zinc finger nucleases when the donor template DNA is present? Option A, non-homologous end joining, which leads to the targeted mutagenesis. Option B, homology directed repair leading to gene editing and site specific integration. Option C, error prone repair resulting in the random insertion of nucleotides. And option D is translation synthesis, which bypass the DNA damage without repair. So in this case, they have mentioned that donor, yeah, you both are right. So whenever donor template DNA is present, it always undergoes homology directed repair. Then, with the help of which it can lead to gene editing or site specific integration. Okay, so next question is, which of the following statements about the CCR5 chemokine receptor is true? So CCR5 is a receptor with 5 transmembrane domains and is primarily expressed in liver and lungs. Option B, CCR5 functions solely as a receptor for signaling molecules in the immune system and does not interact with HIV. Option C, homozygosity of a 32 base pair deletion in CCR5 gene, that is del32, confers strong resistance to HIV infection, while the heterozygosity results in slower HIV progression. And option D is CCR5 is expressed only on CD4 lymphocytes and does not have any role in the central nervous system. Okay, so the right answer is option C. We know that this CCR5, this particular protein, it generates a receptor which is expressed on the surface of the cell, but that particular receptor interacts with HIV, due to which HIV can get into the cell. So due to which this B option is not right, because they mentioned does not, but it does interact. And this option C is right, because there is 32 base pair deletion, and if there is deletion, then it leads to the strong resistance to HIV infection, and if there is heterozygosity, then it results in the slower HIV progression. So this is the treatment which is generally opted by different healthcare institutes, so that the HIV means HIV progression can be slowed down or it could be prevented. So homozygotes leads to resistance to HIV and heterozygotes leads to delayed disease progression. And this is the exon 3 where this um, mutation takes place and where the deletion takes place at this particular site. Okay, next question is, which of the following statements about the cervical cancer and HPV is true? Option A, cervical cancer is most common in high income countries with few cases reported in low and middle income countries. Option B, HPV, which is human papilloma virus. So that HPV types 16 and 18 are responsible for less than 10% of high grade cervical precancers. Option C, human papilloma virus is responsible for the more than 95% of cervical cancer cases. And option D, Sankar and associates use the four-fingered compo-ZR-ZFN pair to target the E6 gene of HPV 16. Okay, so right answer is option C, right? Which is human papilloma virus is responsible for more than 95% of cervical cancer cases, whereas they have intentionally kept this statement. So they have used the six-fingered ZFN and not this four-fingered. And but they used it to pair to the target the E6 gene of HPV 16. So maybe during the exam they might change this option. So just read this carefully, that whether it is four-fingered or it is six-fingered. So next, okay, so we'll skip these questions and move to next week. Okay, so in week 6 you have looked into the talents, how they are designed and what are the applications. So first question is, which system is responsible for secreting tail proteins into the plant cells? Option A, T1SS, option B, T2SS, option C, T3SS, and option D is T6SS. Yeah, so you're right, option C is the right answer, which is T3SS, and which is responsible for this particular secretions. And yeah, and this is this is how it looks like, this system. So with the help of this, the effector proteins or the tail proteins can enter into the plant cell. Okay, next question is, which gene is primarily targeted by the PTHX01 tail protein in rice? Option A, OS Sweet 11, option B, OS09 G2910010, Option C, OS suite 14, and option D is OS suite 13. So OS means Oryza sativa, which is the name of the scientific name of the rice. Okay, so I mentioned option C. So right answer is option A here, which is OS suite 11. And yeah, it's difficult to remember, but you need to remember these things. So in this case, OS suite 11, and there are others also, means 13, 14, all of them are used, but in the case of this PTHX01 tail protein, OS suite 11 is the one gene which is targeted. So this is PTHX01. Whereas if PTHX02 comes, then it targets OS suite 13p. And if it is PTHX03, then it targets OS suite 14p. So in this case of PTH01, just from 1 remember 11, in case if you can remember OS suite 11. Okay, next question is, what is the primary function of talents in genome editing? Option A, protein degradation. Option B, gene expression inhibition. Option C, DNA cleavage at specific sites. And option D is RNA interference. Yeah, so you're right. Option C is the right answer. That is DNA cleavage at specific sites. Okay, so next question is, which enzyme is most commonly fused with tails to create talons? Option A, EcoR1, option B, FOC1, option C, BAMH1, and option D is HIND3. Yeah, so you're right, FOC1, because tail itself is just for the recognition, the nucleotides, recognition of the DNA, where it will recognize and cleave. But for the cleavage purposes, we require nucleases. So for the nucleases, in this case as well as in ZFNs also, we have seen that FOC1 endonuclease is most commonly used. Okay, next question is, which model organism was used for generating the SKID models by a talon mediated knockout of PRKDC? Option A, mice. Option B, zebrafish. Option C, rats. And option D is goats. So I think SKID is the severe combined immunodeficiency disorder. Yeah, so you're right. Option B is right answer, which was zebrafish. And there are some papers also related to it. Okay, next question is, which talon assembly method uses the hairpin capping oligonucleotide to improve the assembly efficiency? Option A, flash assembly. Option B, iterative capped assembly, which is ICA. Option C, Gibson assembly. And option D is star protocol. Okay, so now let's see. So right answer is iterative capped assembly, which is ICA. It's clear from the name also. So in this case, to avoid that, because in at each case you are just uh, means assembling the modules. So in this case, I want to add A, B, C different modules to construct the talon. So to avoid that, for example, I want B to attach after A, because otherwise sequence will be disturbed. So to avoid those things, I use a cap, which is a hairpin capping. So these are some other oligonucleotides which will bind and will avoid the binding of B before the A. So if A is not bound, so the cap will bind, it will avoid binding of B to this particular site, so that proper binding can take place. And this type of assembly method is known as iterative capped assembly or hairpin capping oligonucleotides. So these are oligonucleotides which block the incomplete assemblies. Okay, next question is, what is the common outcome of talons targeting the mitochondrial DNA? Option A, HDR based recombination. HDR means homology dependent repair. Option B, NHEJ mediated repair. Option C, rapid degradation of mutated mitochondrial DNA. And option D is activation of nuclear encoded mitochondrial genes. Yeah, so what we have read is that in the case of mitochondrial DNA, talons are generally used for the degradation of mutated mitochondrial DNA, so that mutations can be reduced, and that is what makes talons most important. Because if we compare it with the CRISPR-Cas9, so right now CRISPR-Cas9 is most commonly used, but in the case of mitochondrial DNA, talon is still used other than CRISPR-Cas9. Yeah, and this is what it shows, that in this case these red ones are the mutated ones and these green one are the original proper DNA. So if mutated DNA increases, then we use these mitotelins or empty ZFNs. So empty ZFNs are the zinc finger nucleases which cleaves these mutated DNAs, and in mitotelins these are the telins which act on these mitochondrial DNAs and cleave the mutated DNAs, so that there is elimination of this mutated mitochondrial DNA, and then remaining mitochondrial DNA can further get replicated and increase its copy. Okay, so next question is, which gene was targeted in the talent based knock-in experiments in the rats for studying the glucocorticoid receptor functionality? So option A is NR3C1, option B IgM, option C PRKDC, and option D is CXCR4. So gene that is linked to this glucocorticoid receptors. Okay, so right answer is option A, that is NR3C1. So this was the gene which was targeted by the talons to study the glucocorticoid receptor functions. Okay, so next question is, which mechanism is used to achieve high targeting specificity with tail base editors? Option A, splitting the deaminase into two non-toxic halves. Option B, using full-length DDDA domains. Option C, co-delivery of talons and RNA components. And option D is incorporating a fluorescent reporter. Yeah, so you are right. Option A is right. That means to split the deaminase into the two non-toxic halves with which we can achieve high targeting specificity. Because this, if we use the full-length DDA domain, then this full-length domain is highly toxic. So we cannot use it as a whole, this deaminase domain. So, and then the third option was that co-delivery of talon and RNA components. So tail base editors are protein only systems, so they do not require any RNA component. So this is also not true. And the fourth one was incorporating a fluorescent reporter. So reporter is generally used to track that where the editing has reached and not involved for the targeting specificity. Okay, so next question is, what is the main advantage of using talons over ZFNs for genome editing? Option A, smaller cDNA size. Option B, lower cost and ease of delivery. Option C, simpler protein DNA recognition code. And option D is higher specificity and low off target effects. So in this case, the right answer is option C, that is simple protein DNA recognition code. So in talons, it follows straightforward one repeat, one ways RVD code. Unlike in ZFN, we have seen that it's one single finger or one ZFN motif. So one motif recognizes triplet of nucleotides. So in that case, the assembly is difficult to find those things. But in the case of talon, because one repeat binds to specific to one base, so in this case the assembly is quite straightforward. So that's why it has simpler protein DNA recognition code. And in this case, high specificity and low off target, so it is partially true. But this simpler protein DNA recognition complex is means that is more main advantage as compared to this one. So this is also means advantage only, but not as main as this first one. Okay, now we'll move to the next week. So in week 7, we have looked into CRISPR-Cas9 technology. So we'll answer questions about it. So first question is, what purpose does the CRISPR-Cas system serve in the organisms where it is naturally present? So option A, defense against viral infections. Option B, regulation of gene expression. Option C, repair of damaged bases. And option D is synthesis of essential proteins. Yeah. So defense against the viral infections, and it was first identified in the E. coli only, the bacterial system only, that how it was fighting. It keeps the memory of the viral particle, and then next time when the virus attacks, so it degrades its genetic material. So that was how the CRISPR-Cas9 system was invented, and later on it was explored for the genome editing. So next question is, what is the primary function of tracer RNA in the CRISPR-Cas9 system? Option A, to degrade the viral RNA. Option B, to guide the Cas9 protein to the target DNA. Option C, to repair the damaged DNA. And option D is to synthesize the new proteins. Okay, yeah, so it's the right answer, to guide the Cas9 protein to the target DNA. So if tracer RNA is missing, then it will be inactive Cas9 and DNA cleavage will not take place. Whereas if tracer RNA is present, then it will guide this Cas9 and it will bind to the DNA, and then uh, PAM proximal binding will take place, zipped conformation, and then DNA cleavage will take place, which is the main purpose of CRISPR-Cas9 system. Okay, so next question is, which of the following viral vectors is widely used in CRISPR gene therapy because of its high efficiency of delivery? Option A, lentivirus. Option B, retrovirus. Option C, herpes simplex virus. And option D is adeno associated virus. Okay, yeah, so it is right. Adeno associated virus is the right answer. Lentivirus is efficient for integrating genes into dividing and non-dividing cells, but it is less preferred because of the safety concerns, because lentivirus might not be safe, it could lead to other infections, so due to which adeno associated virus is used. It is low immunogenic also. Next question is, what is the full form of CRAGE in the context of genome engineering? Option A is chassis responsive active genome editing. Option B, cellular recombination and genetic engineering. Option C, chromosome related active gene editing. And option D is chassis independent recombinase assisted genome engineering. Yeah, so you are right. Right answer is option D, that is chassis independent. So chassis here means organism independent recombinase assisted genome engineering. Next question is, what causes Duchenne muscular dystrophy? Option A, overexpression of myostatin gene. Option B, gain of function mutation in dystrophin gene. Option C, loss of function mutation in the dystrophin gene. And option D is loss of function mutation in the myostatin gene. Yeah, so you're right. So if there is loss of function mutation in the dystrophin gene, and when we were talking about this, then we also read that that particular loss of function can be brought about due to the exon skipping or exon deletions. So there are multiple exons, not just one specific exon which get deleted or skipped. So many other exons can get skipped, due to which the frame gets changed. So now if frame gets changed, so there is pre-arrival of the stop codon, due to which the protein is immature, which gets synthesized. So due to that premature protein, shorter protein, it gets degraded by the machinery. So there is loss of the this dystrophin protein get lost. So that is known as loss of function mutation, which leads to this Duchenne muscular dystrophy. Next question is, which of the following enzymes is fused with the dCas9 for the construction of cytosine base editors? Option A, DNA methyl transferase. Option B, cytidine deaminase. Option C, adenosine deaminase. And option D is reverse transcriptase. So in this they have mentioned base editors. Yeah, so you're right. So cytosine base editors, cytidine deaminase. And this is what is shown. So if there is deamination of cytosine, it get converted into this uracil. And in the case of DNA, in place of uracil, there is thiamine. Next question is, the tripartite activator domain, that is VPR, does not contain which of the following elements? Option A, RAX from zebrafish. Option B, VP16 from herpes simplex virus. Option C, RTA from Epstein-Barr virus. And option D is mammalian P65. So VPR contains 3 elements. So which one of them is not present in VPR? So in this, the right answer is option A, that is the RAX from zebrafish. So means this VPR, it is a tripartite transcriptional activator domain. So This was engineered to boost the gene expression in the case of CRISPR based gene regulation systems. So in this case there are three domains which are fused. One is VP64, and which is derived from the herpes simplex virus protein VP16. So VP16 is a protein which contains VP64 domain. Then P65 domain from the mammalian NF kappa V subunit, and this RTA is obtained from the Epstein-Barr virus. So these three domains combine together to form this VPR domain, which is the activator domain. So that's why Rex from zebrafish is not the right answer. Okay, next question is, what are the two catalytic domains of CRISPR-Cas9? Option A is PAM and RUB9. Option B, HNH and RUB9. Option C, HNH and RUBC. Option D, PAM and RUBC. So catalytic domains. Okay, let's see. So right answer is option C, which is HNH and RUBC. So both of them are the catalytic domains present in CRISPR-Cas9. So keep this thing in mind because these are involved in the cleavage. And okay, so let's see their role as well. So this HNH, it cleaves the DNA strand which is complementary to the guide RNA, and RUBC, it cleaves the non-complementary strand. So this is the non-complementary strand, this is the guide RNA. So the strand which is complementary to this orange colored guide RNA, that is cleaved by the HNH. So keep this thing also in mind. Next question is, what role does PAM play in the CRISPR-Cas systems? Option A, it influences the efficiency of DNA cleavage. Option B, it is involved in the RNA synthesis. Option C, it is crucial for the acquisition and interference. Option D is not essential for CRISPR-Cas system function. So you have seen the PAM site in the CRISPR-Cas system, so you need to tell its role. Okay, let's see. So its role is, it is crucial for acquisition and interference. So this is the PAM. And PAM sequences are essential for the Cas proteins to distinguish the target DNA from the self DNA and enable both the spacer acquisition and target cleavage. So that is the role of this particular PAM. Okay, so next question is, what is the role of inteins in the protein processing? Option A, protein degradation. Option B, protein splicing. Option C, inhibition of gene expression. And option D is enhances the protein synthesis. Yeah, so the right answer is option B, which is protein splicing. Okay, yeah, you both are right. So protein splicing. So just this inteins and introns, these are the similar meaning terms. So when we say intron, we are talking about the DNA which contains the non-coding part which do not code for the protein. So it is removed after the RNA synthesized by the process of splicing. So that is known as mRNA splicing, in which that particular mRNA get matured, introns are removed, 5' capping, 3' poly A tail is added, and then it moves to the cytoplasm for translation. But if that particular same unit, if it do not get removed at the level of splicing, if it is still present and it enters into the cytoplasm where the translation takes place and it forms a protein, then from that protein, if some component is removed after the splicing, then that component is known as intein. So that is the main difference between the intron and intein, and that is the role of inteins in the protein processing. Okay, so we are right now at we have completed till week 7, so this is our week 8. So now, as it's already been 2 hours and I've kept the same questions, so I'll not proceed further, otherwise it will take 2 more hours, I guess, to complete the 12 weeks. So if you have any doubts, you can ask, and otherwise I'll share these slides with you and you can go through all these MCQs, and these MCQs were already present in your the week slides which I have shared. I have taken these MCQs from there only, and this revision session was just for the discussion to quickly go through all these MCQs. So we have practiced enough MCQs till week 7. Yeah, okay. So, good luck for your exam. And mostly it will be MCQ based exam. So, just go through all the MCQs and just clear your facts. So, whenever in the exam the question comes, just read it carefully that what they have asked, because many times what they does is that they do not change the question, but they just shuffle the options or they just change the words present in the options. So, just keep that thing in mind. Yeah, and with that, I'll just end this session. I have kept, actually I have made total 416 slides, so that was too much. Okay, so thank you very much. Okay, you have asked some questions. So what was the need for learning all the outdated technologies when CRISPR-Cas9 and its variants is now the main technology for genome engineering? So the main thing is to learn from the mistakes. So why those technologies failed? What were the techniques which could not work out? So even if CRISPR-Cas9 is present, but we cannot rule out the ZFNs or talents. They have their own advantage. They have some works and they are not, means, if we talk about the talents, so as we have mentioned during the mitochondrial, those mutant DNAs when they were removed, so CRISPR-Cas9 is not that much, we're not that comfortable of delivering that into the mitochondria as the talents can do. So in this way, the primitive outdated technologies, they also have some roles, but they fail at one level, due to which we adopt the new updated technology. And we can also learn from reading those outdated technologies that what work they did and where they lagged, because in many cases we have seen that due to advancements, many people what they did is that instead of innovating some new thing, they have just modified the previous existing techniques. Okay, so actually I'll be mailing all of this into that already available drive, but yeah, your I'll just keep your email ID so that I can mail you separately as well. So I've saved it. It's the PDF version. Okay, so you want the PDF version of this particular presentation in which MCQs are kept, right? Or for all those presentations you need the PDF version? Maybe I'll share you the drive link in which I have kept other things, and for this particular slide I'll mail you the PDF version of this so that you can quickly go through this. So in this particular slide which I am talking about today is, so in which all the MCQs which are kept, so these MCQs are not from your current assignments. So other than these MCQs, also go through your current MCQs in the assignment as well. Okay, then uh, yeah, you're right. Okay, so many things, we do not get patent on particular thing. We are not getting patent on CRISPR-Cas9, but we can get patent on a technique, that how it is explored. So if CRISPR-Cas9 is used for genome engineering at one particular way, then we can patent it out. We are not patenting on the and already existing CRISPR-Cas9. We are just getting patent on the technique how it is used. So that is what got patented. For example, some medicines or some other things get patented, but that doesn't mean the ingredients, we don't get patent on that ingredient, but we get patent on the at what composition those ingredients are used. Okay, yeah, so thank you. Okay, so if there are no further questions, then I'll end the session and wish you good luck for your exams. Okay, so yeah, I'll mail you the slides, everything. Yeah, thank you very much, both of you. Bye bye.